So we're going to talk about the AMP trials, the Antibody Mediated Prevention Trial, which I'll say the following, that the work I'm about to describe is in press in the New England Journal. So it's described in much better detail than I can probably describe it right now. And there's supplemental figures that give a lot more information than I can describe right now. There are like 261 AMP slides. I decided that that would not be the right approach. The other point is that part of the AMP study is another piece of work submitted to Science that I won't go into today, that goes into much more molecular detail about like some aspects of the study. Having said that, I'll start out by thanking people. This was a big study because in the end it's a proof of concept study. And Larry Corey, who's the head of the Vaccine Research Center, and I were the overseers of this study. And then the actual Narad Zumagodi and Surya Chapiganti served as kind of clinical principal investigators. There's a leadership group, a lab group, a statistics group, and a community group, all shown on the slide. As if that's not enough, it was an incredible commitment from DAIDS, and especially Tony Fauci and Carl Dievenbach, the Vaccine Research Center, John Mascola, who developed this antibody, the Gates Foundation with Emilio Mina and Nina Russell, and a cast of others on the right-hand side. And then there were just many other people to thank. So I don't want to give the impression this would ever be possible to do a study this big without like a cast of thousands and thousands and thousands and thousands and thousands more thousands and thousands and thousands more thousands and thousands and thousands of more thousands. And these are the site investigators of our many sites all over the world. And yes, it took an incredibly large village just to try and explore what would seem to be a really simple question. Now, I throw in a coronavirus slide just for one reason. All the work we've done in coronavirus antibodies, and it's a lot, would have been totally impossible without all the investment that was put into broad neutralizing antibodies in the AMP that preceded AMP and continued during AMP. So we learned how to do all this stuff with antibodies because we worked on it so long with HIV. I'm glad at a future date to talk about all the work with the coronavirus monoclonal antibodies, which has been impressive, and some have published, much have been impressed, but it all came from what we're talking about with AMP. So AMP started in 2015, but it really, actually, from a scientific point of view, the really most important thing was in 2011 or so, Michelle Nuzenswag, a truly brilliant scientist, and a guy named Bill Scheid, they developed an assay to isolate single B cells from convalescent blood and to study the antibodies made by a single B cell. And that isolation of single B cell technology allowed then this kind of search for what we're calling now broad neutralizing antibodies in the HIV field. So the AMP study is predicated on the idea that BNAbs are derived from HIV positive person. Now these BNAbs can only occur in untreated people. So the idea that we're going to forever make more and more BNAbs is an indictment of the fact that once we treat people, they'll no longer make BNAbs. Because what happens is the BNAbs are a function of about 10% of people whose B cells keep chasing HIV, generating ever more somatic hypermutations until they make antibodies that kill a lot of viruses, but never their virus, because their virus has always escaped. There's no evidence that the BNAb that is generated by the person with the BNAb has ever benefited the person with the BNAb. And once this technology was developed and the structure of HIV was recognized, the search for ever better BNAbs developed. And this is just a model of the virus. And you see that there are many sites to which BNAbs have been made. By far the most popular idea has been to attack the CD4 binding site. And that's where we focus the AMP attention, at the CD4 binding site. And the basic idea is that there's a lot of viruses out there, and if you had an antibody that could see the CD4 binding site at all the viruses, you would have an unbelievably great therapeutic agent that would lower viral load, or a wonderful prevention agent, which is where we're going to go with the antibody-mediated prevention study. To date, really, this is a little bit out of date, this slide, but it's really true that most energy focused on an antibody called VRC01, that's Vaccine Research Center 01, generated by John Muskell and Barney Graham, who worked on this forever. And then the other two big investigators that deserve attention are Michelle Nuzensweig, who made 3BNC117, an excellent antibody, and Dennis Burden, who has a lot of antibodies but has been a little less clinical. VRC01 is an antibody made by a guy followed at the NIH who was never treated, but because there was no HIV treatment at the time he got followed, but he was bled routinely, and so whenever you see a VRC01, 2, 3, 4, 5, 6, 7 antibody, they're all from the same donor over time, where he's extending his antibody. The other thing I can say before we go on is the VRC01 is going to have a half-life. You're going to give it somehow. In our case, we're going to give it IV, and it's going to be eliminated over a few weeks. It is possible to alter the FC portion of the molecule and extend the half-life of the molecule through this FC modification to a much longer acting antibody. So FC mutated antibodies are very popular and are probably important for the future. They're called the LS mutations. So this is a slide that you would think is very compelling, not compelling. This is a slide that would get us into this whole discussion. What a great idea. You know, you're going to have pre-exposure prophylaxis with something. Why not use a natural product, an antibody? And this was an examination in vitro using an assay called the TMZBL assay, where the viruses are set up in the assay, and you're looking at the a concentration of antibody required to kill 80% of the viral strains. And you see on this particular slide, you know, the number of viruses resistant to VRC01 looks pretty good. About 12% of viruses were resistant to VRC01. So you'd expect about 88% or 90% to be susceptible. On this particular slide, you see that BNC doesn't perform quite as well, and that's a little bit of a complicated argument. You'll see the LS mutation. You're longing the half-life, but you're making you're affecting susceptibility and the concentration required to kill the virus. And I've kind of reversed this a little. It's like 5%. Then on this slide they claim 5% are susceptible, and so on and so forth. So you see that on the left-hand side the titer required and for these antibodies. So you think, okay, this is a pretty good antibody, and it was the only one available when we started thinking about this work. So we argue that maybe the MIC might be 1 microgram, and that would kill 80% of the viral strains, or 90% of the viral strains. So it looked like a pretty good antibody. And then work was done to see whether this VRC01 antibody went to tissues that needed to be protected. And what you see is that you can do biopsies and find the drug at different concentrations in the rectum, and similar work was done on the vagina, that this would localize to these tissues. Now, I'll tell you that when you make an FC mutation, actually antibodies localize even better to mucosal tissues. And by the way, this would be important for the rectum, the vagina, and for COVID, it's important for the nasal mucosa. So we have these different mucosas that we try to protect with antibodies. So the next step for AMP was to do some animal experiments, and they were done at concentrations that were predicted based on the in vitro assays. And you could see VRC01 versus a control in a rectal challenge and a vaginal challenge. And you see that the antibody provided really excellent protection against the SHIV challenge using a particular SHIV strain that we knew was susceptible to the antibody. So you kind of create conditions that you're going to win, and we won. And there are many non-human challenge studies, and there's a lot of papers have been written looking at this more recently about different SHIV strains and different antibody concentrations and so on and so forth. It's not something I'm going to go into today, but there's a paper by I think it's in Cell, and I can send it to Christoph, that goes into this in great detail. And on this slide, we show, how much antibody might you need to protect, and then you see protected versus infected. And we came up with the idea that maybe, at least in an animal, if you had 20 milligrams per kilogram, you got good protection. And if you had 5 milligrams per kilogram, things started to fail. So we tried to get a dose ranging that was, and the dose ranging was estimated based on all that in vitro work with that assay. Now remember, when we're doing this work, we have no idea that the assay is telling us the truth. We have no real idea of all the strains that are out there circulating. You're kind of working in the dark in a lot of ways. And working in the dark, you're trying to get your best estimate of how much antibody should we give to people? How should we administrate it? How often should we administrate it? What would be our power calculations, our anticipated success in trying to make a pre-exposure prophylaxis agent? So we came up with this kind of a slide that shows these overlapping concentrations. We started making the argument that if you had 100 micrograms per milliliter of antibody in your blood, I guess if you had 100 micrograms at the inception, at the first 4 weeks, you had 100 micrograms per milliliter of antibody in your blood, which would be with the 30 milligram per kilogram dose, you'd get pretty good protection. And then this would wane. And as it waned, at the higher concentration, you'd still have a lot of antibody in blood. And at the lower concentration, at the 10 milligram per kilogram dose, you'd have a lower concentration over time. So we came up with this idea that, okay, the two doses that would be of interest would be 30 milligrams per kilogram and 10 milligrams per kilogram. Now, then the next challenge, there are two other challenges in these clinical trials. That one was the only way we could do this was IV, and people thought it would be impossible to do a large trial IV. But we said, look, that's how we're going to do it. The Gates Foundation was very committed to sub-Q administration. This antibody was not potent enough to give anywhere near the amount sub-Q that would be necessary. So we were never going to give it sub-Q. We had to believe that people would take the drug IV. And then we also had to start worrying about another problem. What about, we're giving an antibody that we have no idea if it's going to prevent infection. What about in parts of the world where Truvada, the antiviral, could be used as pre-exposure prophylaxis? What do we do about Truvada? So to get that off the table, what we did was we made Truvada available free for people who wanted to take Truvada, believing that that they would only take it for brief periods of time and take it irregularly. And then we had to have a whole Truvada blood spot measurement system to see how many people in our study were going to be taking Truvada. So we had a dosage, we solved the Truvada problem, we decided we're giving it IV. And the main research questions were, if we gave this stuff, this antibody every 8 weeks, could we prevent the acquisition of HIV? But in reality, this is kind of a slightly different question. What we kind of anticipated was that the highest concentration, we should only see breakthrough of resistant viruses. And then as time went on, we would see breakthrough of sensitive viruses because the concentration would drop. This included a fundamental misunderstanding of our ability to use phylogeny and a molecular clock to understand when infection occurred. We thought we were so smart we would know everything about the day the infection occurred. I can promise you that's proven not to be true. So could we prevent HIV acquisition? Would susceptibility of community viruses influence the preventive efficacy in places where there are more resistant viruses? You see this stuff fail. And most importantly, this was really, in many ways, all about the vaccine field. Could we tell vaccinologists how much of an antibody they would need a vaccine to make to be successful? So those are some of the questions, and there are many more. So I'm going to give you the answers that we accomplished, and I don't know that we accomplished all these answers exactly right, or that everything is defensible, but basically it's like the question, could we prevent acquisition with VRC01? Yes, but, and I'll explain the but. Will susceptibility of the virus that are circulating affect the success of the study? Yes, for sure. And in the end, I'm going to show you that we were just totally wrong about a lot of stuff, and only very sensitive viruses are going to be eliminated. And can we establish neutralizing antibody titers? Yes. And when I say yes, it's yes, and they jive, they agree very well with animal studies. So now we kind of line up all the animal work and we line up all the human work and we see, whoa, the animal guys really told us a lot and we didn't understand how much they were telling us. So this is the study design. We are going to recruit human beings and we are going to say to them, we are going to see you every 8 weeks and we're going to infuse you over 1 hour or less with this stuff or a control, a placebo, saline in a black bag every 8 weeks. And we're going to do it 10 times. And then we're going to keep following you to see when this antibody goes away, is there something we didn't understand? And I'll come back to that question because there's another fundamental issue going on that I'll need to explain. So we're following people then for 104 weeks. So it's a long study. And this is the sites. There's 2 studies really. There's a men's study and women's study. This is our women's study conducted in sub-Saharan Africa. You'll see that there were 642 people, women who got the 10 milligram dose, 645 who got the 30 milligram dose, appropriate placebos, and you'll see the distribution of how many sites enrolled, how many people. And then we did at the same time, with the agreement, we could pool our results, which might in many ways be stupid. We could pool the men's study, which are going to be men who have sex with men, and they're going to be in Peru, Brazil, Switzerland, and the United States. And you'll see we enrolled 2,699 men, and you'll see the distribution and the sites and how many they enrolled. So now we have this giant study. And the thing I'll tell you that's amazing is that people who were in the study loved being in the study. So we had like a 98% retention. So nobody dropped out. Everybody showed up for their infusion. So we had just remarkable retention. And it led me to believe when we started our COVID work, hey, you can give drugs IV and people don't really mind it. And doctors, physicians in practice long ago got away from the idea that they could give an infusion of something in their office. But in point of fact, that probably is going to come full circle because people like getting these IV infusions. And we have a whole behavioral team that has analyzed, well, why do they like these infusions so much? And so I'm going to show you consistent evidence that the sensitive viruses are sensitive and resistant viruses are resistant, that the TZMBL assay works to help discriminate what's going to happen in the results of the study. And I might as well show you the results. So here's what happened. It's like, okay, we're giving the drug and we're measuring, we're looking for kind of breakthrough viruses. And what we're going to see is if you look at now on this slide, we have the efficacy and the sensitivity of the virus to the antibody in a test tube. And what we find is that if the antibody is if the virus is very sensitive, we get really great protection. We don't get breakthrough of those viruses that are less than 1 microgram per mil or less, but there's a very sharp cutoff. And once that virus exceeds 1 microgram per mil, it is going to break through and cause infection. There's like a little weirdness in the beginning where kind of prevention is seen for a few weeks and we don't completely understand it. But if you get above 1 microgram per mil, there is a breakthrough. And that was observed at the 10 milligram dose and at the 30 milligram dose. So the lower dose gave us as much protection as the higher dose in terms of prevention of infection relative to the control. And on this slide, it just shows you kind of now like a more statistical analysis showing that you really got pretty good preventive efficacy for very sensitive viruses and you got really very poor efficacy for viruses that are more resistant. And that will lead us to like, well, what happened to the overall study? So this is the paradox of the study. Success at some level of thinking of proof of concept as we understand it today, and failure as a prep agent. So the overall, VRC01 pool versus control, the overall efficacy was 26.6%, which is not an acceptable efficacy. But the efficacy against those very sensitive isolates was very great. So the first couple of slides say it works, and then I just showed you a slide that said, hey, this stuff doesn't work. And really, there are a bunch of different possible conclusions, none of which are eliminate another necessarily, but the most important thing is the math of what we found in the study. We had assumed that the concentration of antibody that we were dealing with would give us much greater protection than we observed. So the number of strains in the community that could have been eliminated at the 1 microgram dose was only 19%, 23%, is 4% or 6% at less than 0.3 micrograms per mil, and it was 23% at 0.3 to 1 micrograms per mil, and then everything else is resistant to the concentration that we're achieving in terms of understanding the study. So we would never have the power for this antibody to be efficacious when everything is defined by its ability to eliminate viruses that we're calling sensitive at the concentration achieved. So, so it's kind of like you have to reassess the AMP assumptions. So I'm going to actually read this. It's like, based on the global panel of isolates that were assayed before we started, we had thought 70% of the strains would be sensitive to VRC01, believing that all we needed was less than 10 micrograms per mil. That assumption proved wrong, obviously, completely. Our ability to eliminate strains was at 1 microgram per mil, and there weren't many of those strains circulating. In the AMP trial itself, 73% of 64 isolates of the control group exhibited ID80 less than 10, so the predictive panels were okay. So in other words, those things were circulating, but less than 30% of the viruses were sensitive to less than 1 microgram per mil. So we observed only transient protection from anything greater than 1 micrograms per mil. So we were off by a whole log in our assumptions about this study, and which explains why we think the general idea that a BNAB works is true, but the idea that we were right about the concentration is untrue. The other thing that was untrue was our ability to try and understand the timing of infection, like what day did people get infected. I won't go into great detail with that, but I can tell you that all of our kind of assumed brilliance about antibody seroconversion and molecular evolution of isolates that we collected and such all proved to be less than satisfying, to say the least. And so this is just a slide that shows how prevention efficacy declines with quantitative IC80, and it's already been shown to you really that as the isolates become more resistant, regardless of which study, the men's or women's study, the ability to prevent infection wanes. And this is true of the 10 milligram or the 30 milligram dose. So even at the higher dose, we could not deal with variants, molecular variants greater than 1 microgram per mil. Now, what I'm not showing you is like a ton of other stuff, like the founder viruses. You know, we spent a lot of time worrying about founder viruses. We've really improved our ability to understand, are there signatures of the viruses that break through? What's the true number of founder viruses using more modern technology? Lots of that stuff exists. It's not being shown to you today. And this is just the same kind of idea. The preventive efficacy over time is not great unless you're less than 1 microgram per mil. If you have an isolate less than 1 microgram per mil, then over the entire course of the study, we would fail to see any substantial number of those infections. But once you're greater than 1 microgram per mil, we would see infections and not have efficacy. And as you would predict on this kind of bottle plot, as you would predict, viruses from VRC01 cases are from the people who receive antibody, we are going to recover viruses that have greater resistance than from the control group, because we're putting selective pressure on our experimental group and eliminating the more sensitive viruses. So we have 2.4-fold greater resistance among the experimental group than the control group because of the selective pressure that we exerted. So in summary from this, and again, this is like just kind of a rapid summary, leaving plenty of time if people have questions. We measured the autologous and neutralizing viruses in vitro susceptibility to the antibody VRC01 without any LS mutation in 162 of 175 endpoints. We showed that the in vitro viral susceptibility of BNAbs is the predictor of prevention efficacy. It didn't matter about the clade, it didn't matter about sex, it didn't matter about the rectum or the vagina. What mattered was that there was so much antibody in the blood, and it would only deal with viruses that were particularly susceptible. Now, I'm going to come back to, when I'm done with another concern about the whole study, and explain it in a little more detail. We showed that transition from sensitive resistance is very sharp. That is, once you're greater than 1, it's like monotonic. And we believe the TMZ-BL assay does in fact offer some degree of prediction. Now why is this important? Because Michel Nussensweig, a quite brilliant scientist, believes that this assay is not appropriate. He believes this assay uses a cell line in a particular way. Michel isolates monocytes freshly, and he believes the results are different if you infect freshly isolated monocytes compared to a cell line. The problem is freshly isolated monocytes are hard to get, and they vary. Each donor then varies a little bit. So this kind of conflict about what the right assay is a kind of a sub-theme of this whole plot. So conclusions are that IV monoclonal antibodies directed against HIV and SARS-CoV-2 are very well tolerated with no special side effects. Now, why is this important? In the United States, you're allowed to advertise on television therapies. I don't think that exists in England. And every night, there are an hour of advertisements about monoclonal antibodies for breast cancer, psoriasis, Crohn's disease, you name it, there's an antibody. And those commercials have a sidebar at the end of, and by the way, you might have unbelievable, terrible side effects from this antibody. You might get tuberculosis, a fungus, a lymphoma, you might become an obsessive gambler. I mean, whatever, it goes on and on and on. And one thing about physicians or healthcare providers is they kind of have to understand that monoclonals directed against a pathogen are really different than monoclonals directed against the host. So no one's ever to date, the only monoclonal available directed against pathogens right now that's commercially available is for RSV, and it's rarely used only in children. So as we move in COVID and in HIV to monoclonals for treatment, we're going to have to re-educate that these are directed against the pathogen and not the host. AMP trials proved a concept, but did not provide a product, and it was never intended to provide a product. And now we're at a juncture. I'm going to tell you the juncture, but then I'm going to come back to another theme I want to explain. The juncture is, so we proved the concept that BNAbs have the potential to make a contribution for prevention and the potential to be adjunctive for therapy and maybe even for cure. But for prevention, you've got to decide, can you make a BNAb combination that will be competitive with small molecule antivirals, including cabotegravir, that's going to be approved for prevention every 8 weeks, islatravir, that's a pill that will go out for once a month, lenacapavir and other capsid inhibitors that are 6-month sub-Q injections. Can monoclonals be competitive? And it's my belief they can only be competitive if we had a combination that was really, really super duper, because one antibody is never going to be enough, and they have to last 6 months or a year. They would have to be ultra long to have any value in the prevention field. And in fact, obviously, we're not drug developers per se. We've met with the various companies and with the Gates Foundation and NIH. We've come to the conclusion that we've done a good job, and that's narcissistic. We've tried to bring things this far, and now we're weighing, well, what's the really next best step? Now, thrown into the middle of this was COVID. And so all this knowledge we have about monoclonals and HIV got translated to COVID. And so both Lilly and Regeneron got out of the gate with monoclonals that we've already proven to be really quite effective at reaching concentrations in the nasal mucosa sufficient to lower the viral concentration in the nasal mucosa by at least a log and sometimes 3 logs. So we already have permission to use these drugs therapeutically. We're about to get permission to use them prophylactically. So kind of, it's not like this HIV work, which was so intensive, really taught us how to do this IV work with COVID. And so I'm thankful to HIV for that opportunity. Now I'm going to slow down and tell you one other thing. So now I've told you all this, but there is another thing to worry about. And I have to kind of now move to cabotegravir. While we were developing VRC01, we were also developing cabotegravir. That is the Prevention Trials Network. And the cabotegravir is an antiviral agent. We inject IM with 2 injections every 8 weeks to men and women. And we reported and have submitted to the FDA, or are in the process of submitting to the FDA, that cabotegravir is superior to the other PrEP agents available, such as Truvada or Descovy. Mostly because you give an injection, you don't need to worry about people taking your drug. But one of the things we learned in our work in cabotegravir was how much, when you give cabotegravir, you compromise seroconversion. So somebody might acquire a virus in the tissue that's hiding in the tissue and won't pop out or won't be detected unless you use an ultra-sensitive blood assay to see like a single copy of virus in the blood after you've received your cabotegravir. This can happen because you've suppressed replication to the extent that only much later will the virus surface when you stop the drug, or in rare cases, if the virus becomes resistant to cabotegravir. At the CROI meeting on March 9th, my colleague Dr. Landovitz will present all of our results as they relate to this issue of how you know what's going on with people who've received these long-acting agents like cabotegravir. Why is that important? Because as we saw these cabotegravir results, it wasn't rocket science to start worrying that the antibodies would have the same effect. That is, that we might be giving antibody and suppressing replication of HIV in the tissue, but only when the study is over can people seroconvert and can the viral infection blossom. We don't think that's going on to a large extent, and we're doing lots and lots of experiments to prove that's not the case, but it is something to consider that anything we use that's very effective and suppressive that we think has prevented infection might mask infection, at least for some period of time. And any PrEP agent we use, whether it's a pill or an injectable agent, has the potential to make it difficult to understand seroconversion and to recognize infection. Why is that important to us? Well, for the antivirals, we don't want to leave smoldering infections untreated because that's one way to generate resistance. So a lot is still going on with our AMP study, and a lot still going on with our cabotegravir study, which we're done, and we're seeking approval for prevention, but we've learned a lot, and these 2 studies are complementary in terms of the teaching. I've thanked everybody. As you saw, I think there were at least 1,000 people involved in the AMP study. We especially want to thank the Gates Foundation, NIH, the Vaccine Research Center, for their kind of really unlimited support. I would also say, in all candor, well, I like taking credit for everything, Larry Corey, really the head of the Vaccine Research Center, of the Vaccine Trials Network, was a huge leader in this study and helping to organize so many people. And I would also make the point that the lab people, Carolyn Williamson, Lynn Morris, Jim Mullins, Ron Swanson, many lab people did so much work to make these results possible. And lastly, Peter Gilbert has a huge team of statisticians that are really smart that analyze this stuff 1,000 different ways.